and we are really thrilled to have Dr. Matthew Demczko, who is going to talk about mitigating risk and next-generation sequencing. But before I introduce him, let me mention some housekeeping issues. So the instructions to obtain CME credit are on the screen here. We also have little flyers floating around where you have all the instructions, and we can also email you instructions if you provide your address on the sign-in sheet. Our June 14th ID series, so the next one will be on June 14th, will be Dr. Sean Hennessy from University of Pennsylvania, who will talk about pharmacoepidemiology and academic perspective. And do not forget to register through the Christiana Care portal, and the full ID series schedule is posted on the ACCEL DE-CTR.org. And finally, our June 6th Tech Talk, so June 6th, is Dr. Xiaoke Zhang from George Washington University, who is going to talk about non-parametric covariance function estimation for functional data. So very exciting topic also on June 6th. So I'm really excited to present Dr. Demczko here. Dr. Demczko is attending physician in the Department of Pediatrics in the Division of Diagnostic Referral at Nemours, and also the co-medical director of the Nemours Kinder Clinic, which serves the Amish community of Southern Delaware. He's a clinical assistant professor of pediatrics at the Sidney Kimmel Medical College of Jefferson University. He got his MD from Jefferson Medical College and his pediatric residency at the Sidney Kimmel Medical College and at Nemours DuPont Hospital for Children. His current research is funded by an ACCEL CTR pilot grant, and his research efforts, as well as his clinical efforts, involve the care of medically complex children and children with rare disease. He has had multiple awards, the last one being Faculty Educator of the Month from Nemours in January 2018. So we are really excited to have you and listen about the clinical work and the research work that you are doing today. Thank you so much.
Thanks, Claudine. So thank you very much for having me. I'm really excited to be here. I have not been back to Christiana since I completed my residency a few years ago, so it's good to be back after having spent so much time here during training. What I hope to do over the next 40 minutes or so is kind of tell you a little bit about what we've been trying to accomplish in Southern Delaware, specifically with the Dover Amish population. And thankfully, through the help of ACCEL, we've been able to sort of start an interesting research project collaborating with some folks. And so hopefully we'll get a chance to talk a little bit more about sort of the work we're doing and how that will hopefully improve kind of the clinical care for this population and the children of the community. I do not have any financial conflicts. I will admit I do receive baked goods, produce, and free lunches from our families in Dover, but I don't think I need to disclose that. So I want to start with a few stories, if I may, because when I tell people that I do this work, the two questions that generally come up is, number one, how does one get involved with working with the Amish population? And two, why is it important to work with the Amish population? What is it about the Amish population that makes it different, that makes them sort of benefit from different types of precision medicine? So when I was a second-year resident, I was rotating through our PICU, and I met a young boy by the name of John David. John David was a young Amish boy who was really quite sick. And when I met him, he was unfortunately just under 2 years of age and near the end of his life. He had had a long, tenuous course. He had met with multiple specialists throughout Nemours. And no one really knew what was going on with him, myself very much included in that. John David, I didn't know at the time, but I would come to learn, lived in the Dover Amish community. And his family would drive up to Nemours 2 to 3 times a week, each time hiring a driver. So a driver from Dover to Nemours is anywhere from $80 to $100 round trip. So they were making that trip about 3 times a week. And the family did not have insurance for the majority of their John David's life, and so most of what they were paying was out of pocket. I seemed to kind of run into John David throughout the rest of my second year of residency. So when I left the PICU and went to the GI service, John David happened to pop up on the GI service. And then when I went to the hematology service, sure enough, there John David was as well. I didn't think much of it at the time. But what I do remember recalling each time was just, I felt a feeling of unfairness. I felt that there was just something that we as a health establishment weren't doing for this family. The fact that they were paying so much money out of pocket that we really didn't know what we were doing, we didn't give them answers. And unfortunately, we didn't help really prolong John David's life. So John David died shortly after his second birthday at 25 months. And at the time, we did not know what ultimately he succumbed to. And sort of I left and kind of went about residency, not knowing sort of what was to come down the line from his story. So in the third year of my residency, I was approached to take on this endeavor to help the Dover Amish community. And I'll be honest, at the time, I didn't know what I was getting myself into. I was not someone who excelled or had any expertise in biochemical genetics, but the more I looked and the more I read, the more interested I became. So as I was lucky enough that Nemours sponsored me to go to the Clinic for Special Children, which is a medical home in Lancaster County in Strasburg, PA, who've been doing this type of work for the last 30 years and allowed me to train with them and learn about the different conditions and genetic disorders that affect the Anabaptist population. In my preparation for that fellowship, we'll say, I came across the story of these two children. So the individual on your left is Mark. So Mark came to the clinic in 2003. He was born in 1998. He was 5 years old at the time. And at that time, he came with a host of neurologic symptoms. He had pretty severe developmental delay, intellectual disability, seizures. And at the time, he had seen many, many specialists, countless specialists out by where he lives near Pittsburgh in Somerset County, and had accumulated a huge medical debt trying to figure this out. And ultimately, no one was able to successfully tell what he had, diagnose him. He came to the clinic, and through some interesting genetic testing studies that they had done, they identified him as having severe MTHFR deficiency. So many of you may know that MTHFR deficiency affects the way that our brains metabolize methionine and specifically use different methyl groups in the different functioning of the neuronal cells. And so he was essentially cerebral methionine deficient. There's a treatment, an over-the-counter treatment called betaine, which is a powdered mixture. It's about 60 cents a day that can bypass the pathway that's mutated in MTHFR deficiency and actually provide methionine to the brain. And so when he was diagnosed, he got started on betaine therapy. Betaine, unfortunately, was not helpful for him because he was so far along in his illness. However, what the clinic staff realized was that in this small community of Somerset County where he lived, the carrier frequency for that mutation was 30%, meaning that 1 out of every 50 babies that was born in that community would be born with the same severe MTHFR deficiency that Mark had. This led them to work with the team at University of Pittsburgh to try and develop a newborn screening pathway, a newborn screening test to identify this, because they presumed that if you get started on therapy early on, you would negate some of the neurologic sequelae that unfortunately Mark had succumbed to. In 2004, they were able to do that, and it became a part of the newborn state screening program, which now is available in all 50 states. The very first patient to get picked up for severe MTHFR deficiency is the young lady you see on your right. That's Ruth, and that's his sister. Ruth was started on therapy 2 weeks into her life, and now is a normal, healthy, I think she's 15 by now, normal, healthy 15-year-old without any neurological sequelae and developing normally. So it was this type of work of sort of being able to investigate, make a diagnosis, and then ultimately, most importantly, lead to a treatment and improve a clinical outcome, which really got me excited about this work. So fast forward to today, and this is what I hope is kind of our Mark and Ruth story. So some of you may have seen this story. The Delaware News Journal published a story of one of our young patients named Grace Miller. Grace Miller is this young little girl here. She is right now 33 months of age, and we have been following her since shortly after her birth when she was delivered in our NICU. She came to us with we were consulted on her, one, because she was from the Amish community, but two, because she had a wide variety of symptoms and multi-organ dysfunction that was not really, no one could really make the diagnosis. When we came to see her, we sort of talked with our partners at the Clinic for Special Children. And ultimately, knew fairly quickly that she had a mutation of what was called YARS, so Y-A-R-S. YARS mutation is a gene in the tyrosine tRNA synthetase. So essentially when your body is making protein, that last step when a tRNA puts an amino acid onto a growing peptide chain, the protein that's sort of the helper in that process sort of puts the tyrosine where it needs to be. And she carried 2 detrimental mutations in that and ultimately sort of had a multi-body dysfunction because of that. The only reason that we knew of what Grace Miller had was because she has the exact same condition of what John David had. So John David, the month before he went to, unfortunately he passed away, his family took him to the Clinic for Special Children in Lancaster, and through a really elegant family exome study, discovered this pathologic mutation, and through a number of collaborators was able to do functional testing to show that this mutation was not only compatible with life, but would cause severe multi-organ dysfunction, the likes of which John David experienced, and now Grace is undergoing. Grace is thriving right now. She, as you can see, she has her cochlear implant on. That's her latest addition. She's now learning how to speak, and she is really doing quite wonderfully. And I put up here, this is the family tree for the family. So the arrow is our proben, and that's John David, who we first met. I don't know if this works. Right here is Grace Miller. And then you can see there's actually 2 other children who have the same condition, and 3 of which unfortunately passed away before the diagnosis was ultimately made. So what we hope is that by this type of understanding and genetic research, we can sort of lead to improved clinical outcomes, like the one that happened with Mark and Ruth, and now the one that's happening with our patient in Southern Delaware. So briefly, just to kind of go over what I'd like to talk about today. So we're going to talk about a little of the unique historical events that kind of put the Amish and Mennonite population at an increased risk for inherited disorders. Because there's a lot of information out there of people thinking why they have such genetic disorders, and we'll just kind of clear up some of the misunderstandings that may be out there. We'll also talk a little bit about the Dover community, what we know about it in terms of its history and also its genetic makeup. And finally, we'll talk about our study and what we're looking to do and how we hope that it might be able to help future generations of children within the population. So I would be remiss if I didn't put a picture of these 2 gentlemen on a slide when talking about the Anabaptist population. So the gentleman on your left is Maynard Simmons. So he was in the late 1500s, around 1593, started to pull away from the Catholic Church because he believed in the idea of adult baptism. So Anabaptist means literally adult baptism. You can imagine at the time that was not thought of positively among the Catholics and Protestants of the time in Europe. And so they were persecuted quite severely for it, such that they had to retreat to the mountains of Switzerland, into the Swiss Alps. And it's there where they started to farm, and it's there where they started to practice their religious ceremonies within their homes, which is a tradition that's still continued today by the Amish and Mennonites. So as you can imagine, Mennonites came from Manos. And then in the 1600s, this man on your right, Jacob Amen, broke off another sect, and his belief was in very much more strict, pure doctrine, purity, the idea that for excommunication for those that aren't believing in the faith, and his followers then became to be known as the Amish. They also were persecuted, and they also fled to the mountains of France and Germany. And the persecution at some point got so bad where they decided to make the trip over to America. So at the time, William Penn was preaching his theory or his doctrine of religious acceptance, religious tolerance. And so they made the trip across the Atlantic to come to America to sort of settle, especially in Pennsylvania, but in the new land. That all is to say that there is an important part from the genetics of the Amish that comes from those historical events. So at the beginning, the Swiss Anabaptists were an incredibly large community that were setting up shop in Switzerland, France, and Germany, as I mentioned. When they came to America, the trip was quite hard, and many of them did not make it. So the actual settlers that ended up arriving in Pennsylvania, especially, were a very small population, just a few hundred. And we call that founder effect, or a founder population. Those founders then became the sort of ultimate genetic diversity for the rest of what the Amish community became. So you can see that after the founders, they began to develop and separate into smaller groups. And because of the sort of effect of genetic drift, so the idea that just by happen chance, certain alleles become more prevalent in the community, some tend to go away, that in addition with this bottleneck led to the sort of genetic homogeneity that you see in all these Amish populations. Also helping the fact was that they were geographically restricted. So each of these communities stayed in very certain areas and locations, which didn't allow for more genetic diversity to come into the community. And there was no inward migration itself. So as you know, the Amish and Mennonite cultures are very strict in terms of their family makeup. And so there aren't many people coming into the faith. And so the genetic diversity then also does not come into the faith as well. So the collection of these sort of four effects have led to the unique genetic architecture that the Anabaptist population has today. And what's interesting is each of those purple dots can represent the different settlements that we see now throughout America. So the Ohio Amish are very different from the Lancaster Amish. And the Lancaster Amish are very different from the Indiana Amish, such that some conditions that you see in one, you absolutely will not see in another condition. So each clinic, which we'll talk about in a couple of slides, is treating very, very different genetic disorders. So this is just sort of a breakdown of kind of the specifics as it comes to the Mennonites. And you can see each time along the way, there's a new bottleneck effect. And each time there's a new founder population that then develops and grows and expands. And so at each of these points, you can understand that the genetic diseases you see in each conference would be very, very different. So I like this model. I don't mean it for it to be oversimplistic, but I think it really kind of hammer home the point where if you have a bag of marbles with each of those marble colors representing a different genetic allele, if you ultimately just randomly pick out samples and then allow them to repopulate or regenerate, your proportions are going to be similar. You're going to have about the same numbers of each allele. But if you undergo a bottleneck effect, essentially you only pick out a very few number and then allow that small collection to repopulate, you see there's going to be a significant reduce in genetic variation. So if you take, for example, that the green dot may be the mutation or the allele for maple syrup urine disease, it's now at a significantly higher proportion than it had been previously in the original population. And similarly, if something such as glutaric aciduria is our yellow dot, now it's no longer present, so you don't see that condition anymore. So this is exactly kind of explain why the genetics of the Anabaptist population is what it is today. Because of that, these are the unique disease instances that we see in the population. So maple syrup urine disease is a condition seen in the Mennonites. In the general population, we see it very rarely, 1 in 185,000 live births. In the plain population, and specifically in the Mennonites, we see it in 1 in 380, which is really pretty fascinating. Similarly, cartilage hair hypoplasia is a skeletal dwarfism that's also associated with an immune defect. Very, very rare in the general population, but in the Amish, it's 1 in 1,300. So you can see there's definitely an increased risk for this population outside of what we would be expecting from the general population. So this is the Clinic for Special Children. This is in Strasburg, Pennsylvania, in Lancaster County. The clinic has been doing its work for 30 years now. It just celebrated its 30th anniversary this year. And I think what's most striking to me about the clinic, aside from the fact that it's situated in the middle of a cornfield, and those are not props, those are real cows that wander about the field on a daily basis. It's quite fun when the cows escape and the staff has to go out and try to round them back up. But I think what's most fascinating is that with the work that they've done, they really truly understand the genetic factors at play within the Lancaster population. They understand their population such that when a child walks in the door, sometimes even when a child is just born to a midwife, they know what they're looking for and they know what to test for. And for some of these incredibly rare and fatal disorders, they can have an answer in a matter of hours, in 2 to 3 hours sometimes, and be able to start treatment before symptoms even begin. And it comes from all of the work that they've done over their last 30 years. So the reason that they are where they are is because there's a need there. So this is an old map, but this looks at the country and specifically the higher percentages of where the Amish live within the United States. And you can see one of the highest pockets is, of course, in Lancaster County. There are also a number of kind of hotbeds, would you say, in Indiana and Ohio, even in Wisconsin as well. And not surprisingly, that's where all of the clinics who are taking care of the Anabaptist population are located. So ours being down in Delaware, and then there's 2 in Ohio, 1 in Indiana, 1 in Wisconsin, and then of course the clinic in Lancaster County. And again, these clinics come out of the need to help these populations. Generally, the Amish and Mennonite communities are underserved when it comes to medical access to medical care. And so these clinics provide them a geographically convenient location to get the type of care that they need, which often is involved with sort of understanding the genetic diseases that they're at risk for. So going and thinking about our community in Delaware, we have a unique community in that it's fairly new. So as you may recall, the original kind of Amish Mennonite settlers came over in the early 18th century, 1720 to 1730. Our population wasn't founded until about 1915. That's because most of the families who come to Dover have come from other areas. So most have come from Ohio, most have come from Wisconsin, and then a scattering of others have come from Western Pennsylvania, actually some from Montana, and some from Michigan as well. So there's sort of an amalgamation of different families and different settlements that have come to our community here in Delaware. It's still a pretty small community just by size. So this is sort of a general map of the area. It's about 15 by 15 square miles that we see families from. Just west of Dover, areas like Pearson's Corner, Hartley, and we think that there's about 2,400 individuals in the population, and it's based upon church directory information that the population has shared with us. We think that's spread into about 320 families and about 11 different church districts. So as I think about sort of the care that provide to the Delaware Amish, this picture seems to kind of summarize the way that I think about it. So there's clearly a long road ahead. And if you notice at the end of that road, it sort of turns off. And I'm not quite sure from the picture where it turns off to. And that's somewhat how I feel about the Delaware Amish. The reason being is that because they come from so many different settlements, we don't know what genetic diseases they are at risk for. They're probably at risk for a lot of the Ohio diseases. They're probably at risk for the Western Pennsylvania and Michigan and Indiana. They may be at risk for disorders that we haven't seen before. And so there's definitely a feeling of unknown as we start to go about this, because we really know very little about the genetics of this community, whereas the clinic in Lancaster has had 30 years of experience to try to figure out what they're kind of getting themselves into. So we went out to kind of ask this question as we started our clinic in Dover. Is there a way to better prepare for what's about to walk through the door? Can we get to a point like the clinic has done over the last 30 years, hopefully not take 30 years, but can we get to a point where we know when a child walks in the door, when a child is born, what to test for, what we're looking for, and be able to intervene to improve their quality of life and maybe in some instances save their life? So this was our hypothesis. So we predicted the Dover Amish community represents an isolated founder population with a unique pattern and distribution of pathogenic variants or mutations that can be detected early in life at low cost with high accuracy using a customized next-generation sequencing platform that can provide clinical benefit. I think that last sentence is really, those last few words is really the most crucial part, because understanding the genetic architecture is fine, but unless you're using that information to improve the outcome, improve the health of the community, then it's really not a fruitful effort. So thankfully for me, when I started this job, there was already a collaboration that was ongoing that I have been able to benefit from greatly. So the clinic in Lancaster has been partnering with Nemours for a number of years on clinical care and research care. And Archer is a company that has started producing customized genetic screening panels. So the ability to screen for to sequence multiple genes in a very short period of time using next-generation sequencing. And Aaron Krauge, who's the director of computational medicine at Nemours, has really been instrumental in this, both partnering with the Clinic for Special Children and Archer. And so by the time I had sort of rolled in and asked this question, there was already a lot of work being done to help utilize this collaboration and be able to apply this technology. So we're very grateful that the Delaware CTR and ACCEL program funded our project. We are just coming up towards the end of our first year of funding, and we just received notification that we have a second year of funding to continue this work, which we're very appreciative of. And so these are some of our co-Is that both at Nemours and at the Clinic for Special Children. So these are our study aims. So we have two. So the first one I sort of already have hit on, this idea that using this new technology, this new next generation sequencing panel, we can identify and really characterize the genetic architecture of this very well-described homogeneous population in Dover. But the second aim is to improve the engagement of the community. So the community for the past has told us that most of their care, when they need it, they have to come all the way up to Wilmington, and doing so can be very taxing financially on them. And not only that, but they also feel like that many folks did not understand sort of the unique risk factors that they that they were at risk for. And so our hope is through this study, by understanding this, to improve our engagement with them and help them to understand that some of the genetic testing technology that we're talking about may help them and future generations. And so we're going to do that through some survey data, which I'll show in a minute. So I'm by no means a molecular biologist or a biochemist, so I'm not going to try to explain the specifics of next generation sequencing. But I put this up just to remind myself that this is really the technology that allows us to do this work. So we, in our panel, will be looking for 1,300 different genetic mutations. If we were to do that in the old method of Sanger sequencing, that would take months, maybe even sometimes years, to be able to do that in a step-by-step fashion. Next generation sequencing has changed the game in terms of diagnosing genetic variants because it allows us to do it in a very quick and a cost-efficient method. So without this technology, none of this project would be available. So as I mentioned, Dr. Krauge and her team, with the bioinformatics team at Nemours, has been started on this a few years ago. So when they worked with ArcherDX, the first thing that they did was created a customizable panel for the conditions that they see within the Lancaster Amish. So they looked at 63 control subjects within the population, which the clinic had samples on. And so they all had some sort of genetic disorder. So this was sort of a validation type study. In their first panel, version 1.0, they were looking for 202 different genomic regions of interest that equated to 162 different syndromes that are seen within the plain population. What they found was pretty fascinating in that they found all of the known genetic conditions that that the clinic staff knew these subjects had. But in addition, they found many, many more genetic changes that they were not expecting. So 273 different polymorphisms and small insertions or deletions that were pathogenic or could lead to human disease. They additionally, at the same time, also found 35 copy number variations, which is a unique part to this panel where you can look at both point mutations as well as copy number variations. And they found one interesting chromosomal abnormality that was not anticipated. So in this figure in part B, you see the distribution of the number of variations detected per sample. So it's pretty fascinating to see that in just a small cohort of patients, and with only looking for 202 kind of areas of interest, most patients on average had 5 different pathologic variants within their sample. Yeah.
So the 63 Amish and Mennonite here are adults? Or?
Adults, yes. Yep, that's adults. Yep, exactly. They may, the kind of biobank or the repository of genetic samples that the clinic has been accumulated over the last 30 years. So some of these patients may have come in as children, but at the time of when they were testing them, they all were in adulthood. Yeah.
So they are normal, healthy?
They are, one more time. Not necessarily. So for example, so that's a good question. So in the bottom variant, this is just an example of one of the patients that they screened. So this was a patient that they knew had SMA, spinal muscular atrophy. And so through this panel, they were able to confirm that. So they saw that they had a complete loss of SMN1, the gene that causes the most severe type of SMA. So they confirmed that, but they also found that the same patient was heterozygous for both of these pathologic variants. So one that causes ataxia-telangiectasia, as well as McKusick-Kaufman syndrome, none of which were known before. So this patient presumably came to the clinic seeking care for spinal muscular atrophy. And what they found through this pilot data was that not only did they have the known SMN loss, but also had 2 of these variants that could be pathologic. So from this data, we kind of outlined our project. So we are using panel version 2.0. So where they were looking at 202 different genomic areas of interest or variants in that first panel, we're now looking for 1,300 different variants. About 500 of these variants are population-specific genetic conditions that we know exist in Ohio, Indiana, all the other Amish and Mennonite communities that we collaborate with on that map that I showed you. There is also a large proportion of variants that are presumed to be pathogenic, who may not have been seen either in human disease yet, or that have not been seen within the Amish population itself. So from the 30 years of data that the Clinic for Special Children has, they have an enormous amount of pathologic variants or presumed pathologic variants that have not been seen in human disease. So what this will possibly do is allow the diagnosis of a variant or a novel condition that hasn't been seen before without having to do sort of the retrospective work of going back and investigating the genetic material. This may actually be helpful in diagnosing from a prospective encounter. So our population is 2,400. We're taking a sample of roughly about 300 patients, and those patients will be undergoing two arms to the study. So they'll be undergoing the panel of the ArcherDX panel, again, looking for those 1,300 variants. They'll also be undergoing whole exome sequencing. The reason for this is not only is it allow the Archer panel to be validated, we don't have much concern about the Archer panel needing to be validated, but what it does do is allow us to refine future versions. So for example, if we do whole exome sequencing, we find that there are variants within the population that either have not been seen before in the Anabaptist population or specific to the Delaware community, that allows us to add those variants to the version 3 and version 4 of the panel. We can do this for about $200 a sample. And then, as I mentioned, the secondary arm of the study is to better understand the community's perception of genetic testing. And to do that, we are having all of our participants fill out a 23-question survey to better understand sort of how what they think about expanded carrier screening, expanded genetic testing, and sort of how to make use of the results that we get from that type of testing. This is unpublished data from the clinic that is to sort of hammer home the importance of that second arm of the whole exome sequencing arm. Because many people will say, well, if you're already looking for 1,300 variants, why do you really need to go through the trouble of doing whole exome sequencing? So this is data from an unrelated study that came from the Dover population. And what you can see, the conditions or the gene mutations that are highlighted in yellow are actually pathogenic mutations as listed on ClinVar, which is the kind of repository for pathogenic variants that have not been seen before in any Anabaptist population. So these variants, again, listed in ClinVar, can cause changes in brain development or neurologic sequelae. They can cause epidermolysis bullosa, glaucoma, or type 2 diabetes. So this is the importance, I think, of doing whole exome sequencing in parallel to this, because if we find that these are seen in high frequency within the population, it would behoove us to include those in version 3 of the panel, and version 4, so we know that we're looking for this in future generations of children. This is a little schematic to kind of show, one, that we do a lot of driving, and two, that there's a lot of movement back and forth from home base in Wilmington in order to do this project. So we're in Dover a few times each month to do sample collection. Most oftentimes it's actually at the home of the bishops. They've been very open to us coming into their community, coming into their home, and they organize an event where members of their church community will come, and we will sort of explain the study to them, consent them, and do collection right there. We then transport the specimens back to the biobank at Nemours. So Diana Corral, Marley Gones, and their team have been really helpful in isolating DNA for us and then prepping it for shipment over to CSC in Lancaster, who are ultimately doing the sequencing. They then send the blinded data back to us, myself and Aaron within the bioinformatics core. And then the last step, which I think is an important step, is that we have told the community that we plan to return and hold a large community gathering to sort of explain our findings, to tell what we found and how we hope from a population genetic standpoint we can help their community and what this will lead for better health outcomes in future generations. So this is just a general study design, and we chose 300 samples, which will be taken from 150 couples within the Dover settlement, because we know, based upon calculations for looking for detecting minor allele frequencies, that with 300 samples in a population of this size, we should detect about 99% of alleles that have a minor allele frequency of less than 1%. And to do that, we're actually doing stratified sampling of the couples based upon their last name and their church district. The reason for this is that this is work from the clinic a number of years ago that looked at the rates of variance and as they pertain to different congregations or different conferences, as well as different last names. And as you can see, certain variants certainly match up to certain last names. And so we use this as sort of a way of increasing our detection rate, increasing the amount of genetic diversity that we can pick up within the community. Because if we only did 300 samples from the millers, we probably would not have the same outcome or the same genetic diversity as we would from sampling from different last names and church districts. And so this is sort of our breakdown of last name. You can see Miller is far and away the most common last name within the population, but there are certainly other names that come from different original settlements within the United States that we'll be hoping that people consent to participate so that we can really better understand the genetic diversity. So these are our early results. So we just sent our first 60 samples to the clinic. As you all know, in participating in research, there have always been hiccups and delays that you don't expect. So we finally have gotten the samples shipped, and they are actually on the sequencer as we speak. So we are supposed to get our first results out. I'm hoping tomorrow, or today now, Friday, or Monday, which I'm pretty excited about. Interestingly, the average age of our participants is about 34 years. And 100% of them actually opt to learn about their carrier status for the American College of Medical Genetics 59 reportable conditions. So the ACMG makes it clear that when you're doing broad genetic screening, there are certain conditions that warrant themselves to treatment. So understanding and knowledge of those treatments should be allowed to be passed on to the participants so that they can seek treatment if they want that. So they have the option of opting in to learn about those mutations or opting out. And 100% of our participants thus far have opted to learn about their carrier status for those conditions, which tells me that there's a certain degree of understanding of the kind of role of genetics and the role of anticipatory guidance and seeking medical care. In terms of our survey results, so we found that most of them are only partially completed, which is interesting, and I'm not sure that we know why that is. We have written it sort of at an 8th grade level to try to make it easy to understand. All of the Amish and Mennonite patients only go to school through 8th grade, so we try to keep it an 8th grade reading level. And we're not really sure why they don't answer some of the questions, if it's because they don't particularly feel strongly about it, or if it's just simply because they don't understand the question. So we're looking to try to better understand why the results have been a little bit skewed in that sense. One of the questions we ask is what the prices they'd be willing to pay for this type of kind of expanded carrier testing. And most say somewhere between $20 and $100, with $100 being the most common response. Most of the responses are favorable towards expanded carrier screening, which I'll show you in a second. And then a few, interestingly, have raised concern that carrier screening may disrupt the natural order of things. So I apologize, this probably does not transmit very well through the screen. Essentially, green is agree or strongly agree with, where red is disagree or strongly agree with. So for example, one of the questions asks, expanded carrier testing allows me to avoid surprises at birth or during my child's early life, which over 80% of our participants so far have agreed with. The question of whether it interferes with the natural order of things, this is slightly higher than kind of what we expected it to be, but still up to about 50%, actually 60% disagreed with that statement. And then the last question, so it allows me to make specific decisions about my baby's care at birth or shortly after birth. We didn't have any participants that have disagreed with that. We've had 70% agree or strongly agree. So early results show that the population understands and tends to look favorably on this, but we certainly still have a lot more work to do and a lot more samples to collect. Just for interest's sake, this is the known genetics of the Dover Amish so far. I mentioned the Yarzopathy already, and that is a condition we're actively treating in 2 young girls in Dover and then 1 in Ohio. There's also been a few cases of OTC deficiency, which is a urea cycle disorder and the way our body breaks down proteins. We have 2 forms of cardiomyopathy that we're in the process of doing a large family study on to better understand. There's a number of members with PKU. We've had 1 young girl with SCID, that's due to an ADA deficiency. And then we've unfortunately had 3 young infants born with Cockayne syndrome, which is a progressive neuromuscular condition, which unfortunately leads to death by about 2 years of age. So just to kind of summarize and close and kind of come back to our initial story. So this was, again, John David, this was our index YARS case. So throughout the course of his life, he underwent 14 hospitalizations that totaled 129 days of his short life in the hospital, and greater than 50% of that was spent in the ICU. He had 45 outpatient specialist visits, completely unrelated to those hospitalizations. He had 29 scheduled infusions. He had 5 surgeries, and again, unfortunately died at 25 months of age. With the knowledge that we've gained from his case and sort of knowing the genetic mutation and what we're working with, our 2 current YARS patients together, they've each had 5 hospitalizations each, which include their Grace's initial one after birth. On average, they've seen about 8 specialist visits, and that has decreased significantly now that they've gotten older and we are seeing them regularly in Dover. They've not had a single scheduled infusion. They've had 5 surgeries combined between the 2 of them, 2 of which were elective cochlear implants to allow them to hear. And as I mentioned, they're now both thriving. They're actually 33 and 32 months now, but they're doing quite well. And this is a picture of Grace working with one of our speech therapists at the hospital at Nemours. So we still have a lot of work to do, but one of the projects that we're working on now, as I mentioned, is that we know that there are 2 unique genetic cardiomyopathies in the community that we're hoping to learn more about. So we're collaborating with cardiologists in the Lancaster area as well as University of Delaware to help with better understanding and characterizing that. And then Erin has really been sort of the champion of the partnership between Nemours and Archer DX, and she's been doing a lot of work with her next generation sequencing panels in the oncology population, has really done some really cool work. We'll continue to do that in helping children with cancer. So it's exciting to be a part of that collaboration. So this is, in closing, this is a picture of our clinic down in Dover. So Kinder is the Pennsylvania Dutch word for child. So we are set up within the Dover primary care office at Nemours, and we have kind of our own section to the side with our own waiting room, a few exam rooms that are a little bit more not as kind of neon colored, which is based on what the families asked us to do. We kind of consulted with them a lot about how to design the rooms, and they gave us a lot of great ideas, a lot of wooden structures. We're still waiting for a hitching post for our horses that come in with the buggies. We haven't gotten that yet, but we're hoping to soon be able to have that in the back for our patients who drive in. So just to acknowledge the folks who have been very, very helpful. I've had a great number of support of the folks at Nemours. Stephanie has been our coordinator, as Lauren Bowser at the Clinic for Special Children has. Mike Fox has kind of been my partner in crime with this work and is the other co-medical director of the work we do down in Dover. I mentioned Aaron, and Rob Akins has been a great mentor, as Matt D has. Marley and Diana run the biobank, and then we've gotten a lot of institutional support from Andy Cole, Jay Greenspan at Nemours. They really feel strongly about this work. And then, of course, our funding source, which we couldn't do without ACCEL. So with that, I will open any questions. Yeah. That's a good question. So the question was, can we do anything before birth while the baby's still in the womb? So for the conditions that we know of thus far, probably not. What's been a big hot topic now is the idea of gene therapy and sort of the ability to actually correct a genetic condition, which we're still many, many, many years away from. But in theory, that could be something that we could consider for some of these conditions. But right now, we don't have any treatment options prior to birth. It's usually right after birth, sort of making a diagnosis and then getting a treatment started. Good question. Yeah.
Great talk.
Thanks.
For the adults that you are screening, are you finding anything where they have higher risk for common disease, prostate? And has there been any conversations of more frequent? I guess the second question is access to care for the parents of the kids that you're taking care of. Do you feel that there's primary care?
Yeah, that's a great question. So the question was, are we finding any sort of common comorbidities in the adults that would benefit from early treatment? And if we do, is there access to care for those individuals? So we are definitely finding those conditions. So a part of those 59 conditions that the ACMG says that we can report out are things like familial hypercholesterolemia, diabetes risk factors, et cetera. And so we will definitely find those, no doubt. A part of the panel as well, we know that there's also a specific mutation in ApoB, which causes familial hypercholesterolemia at a pretty high rate within the Amish. So we expect to find that on the panel itself. And secondly, within those 1,300 conditions that we're testing for, interestingly, there's the clinic staff has tiered them. So tier 1 being the ones that we know cause human disease. Tier 2 being the ones who may or we think presumed pathologic. And then third is sort of situation-specific conditions. So, for example, we know there's a mutation that causes an increased risk for muscle disease in rhabdomyolysis in folks who have statin use. And so we know that if that's detected in those patients, it would behoove us to tell them they shouldn't be on a statin because they're at higher risk for muscle disease. So we know that those things are out there, and we know that we're going to find them. The question to access of care is one that I don't know yet. So I know a lot of the families do have physicians. A lot of them have cardiologists. A lot of them have primary care doctors. I don't know if they're a part of like Christiana satellites or who they see, but that's going to be something that when I return to the population and talk about this, I would like to learn more about so that we can start to address that, because I don't know the first thing about how to treat high cholesterol, I'll be completely honest. So having the help of folks like yourself and IM docs would be really helpful. Yeah.
So in the questionnaire that you administered, were there I guess there were self, and were they both men and women?
They were. So they're both men and women. And the way that we've been doing it is that we administer it, we distribute it at the sort of collection event. So when they're consenting, we hand the survey out and we explain it to them and we kind of go over it. And we ask that if they can, they fill it out while they're there with us. So they return it to us with their consent form. So that way we know they've completed it. There are some families who say they would like to take it home to think more about it and sort of really dive into it more. So we allow them to kind of mail it back to us. So we give them sort of an envelope with stamp to mail it. And we've had a few people mail it back, but in general, we find that the people who fill it out right then and there tend to fill out more than the ones who mail it back to us. So we're thinking about, rather than maybe allowing them to do it, but really encouraging them to fill it out in our presence and ask us questions if they have any.
So was it one questionnaire
per participant? Yeah. So if we're screening couples, both members of the couple get it. And it's interesting to see that in a few instances, the couples definitely differ in terms of how they perceive it. Yeah. 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 So the question was, what's the kind of engagement like with the healthcare system? That was one of my biggest concerns when I started this work, when I started this job, is are we going to get buy-in? Are people going to really want to be interested in doing this? And I've been amazed and overwhelmed by the answer being yes. So the families that we have taken care of have been very open to the therapies that we have proposed to them, so long as it improves the quality of life for their child or can help other generations of their children. So I've heard that term a lot, the idea that if this project, if this research can help my children's children or my children's grandchildren, we'll do it. We're interested. And so that's been really exciting and really helpful. There is certainly the component of finance to it. So they do understand that there are things where there are some patients who are on Medicaid, but for the ones who are not, they really have to think critically about whether or not they can afford the treatments that we offer. And generally the community will come to the rescue and sort of help them and join in and distribute funds to be able to do that. But there have certainly been times where they've, talked a lot about the necessity for a treatment based upon how much it may cost. So in general, this population, very open to the work we've been doing, the treatments that we've been offering to them and the research, but that's not always the case at other communities, other Anabaptist populations around the country. Yeah.
Is there any benefit of this?
Wonderful question. So the question was, is there any benefit to this type of study to the general population? I sure hope so. That is, I think, the ultimate goal of the work that we're doing with the community. So because they are these small founder isolated population, they allow us to really understand the genetics very well. But the hope is that not only can we apply that knowledge of genetic conditions to the general population, but somehow be able to apply this model of precision medicine to the general population. What that will look like, I don't know that I know that just yet, because the reason we can do this type of work is because we know the population-specific variants that we have, and all of that changes when you think about the general population and the amount of genetic diversity that comes with that. But the hope is that this type of technology can become more utilized in medicine, so we're being more proactive when it comes to diagnosing genetic conditions than retroactive. Because I can tell you in the work that we do sort of in our clinic at Nemours is a lot of times trying to make a diagnosis after they've seen hundreds of specialists and had $1 million workup. Whereas if we could somehow figure a way to do that preemptively, proactively, we would save a lot of healthcare costs and we would probably improve a lot of quality of life as well. Yeah.
Is this sort of are there lessons from those?
There's not been a study like this done that's been resulted yet, but there's another study that's ongoing right now in the Ohio Amish with a similar idea that they've made a panel kind of specific to their community with many less variants. And I know that there's folks from Wisconsin that are thinking about the same thing. So people are realizing that in these founder populations, this type of technology can be very, very helpful. So it's ongoing, but we don't have any data to say that it's, aside from the pilot data we presented, we don't have any other data to run on yet.
Hey, Matt, this is Erin on the phone.
Hey, Erin. How are you?
Fantastic presentation. I'm great. Fantastic presentation.
Thanks.
And while you were speaking, your first set of data finished processing, and it's all ready to go.
Sweet. That's awesome. Thanks. I'll see you in like a half hour.
Perfect.
Yeah. So genetic testing that is done, that has been developed already, is done in all Amish communities?
So the way that the clinic has designed their workup, they call it the diagnostic pipeline. So essentially when a child walks in the door, the first, obviously with taking a history, if they feel like they know what disorder this is just based upon the clinical presentation, they do targeted testing for that condition. So they know, and in their lab, they can do, specific gene testing for whatever the condition is. If they don't either have a targeting test or targeting testing is negative, they move on to microarray. So essential, looking for large copy number variations that may explain the condition. If that's ultimately negative, they then proceed on to whole exome sequencing. So essentially looking for conditions that they either have not seen within their community to date or conditions that may be novel, that have never been seen before. And they have an enormous number of conditions, novel conditions that they've made a diagnosis of based upon that pipeline. In the paper that they published about a year ago, their diagnosis rate using that pipeline in their population is about 76%, which is pretty remarkable in the world of genetic testing. Most times when you get to whole exome sequencing, the diagnosis rate is probably more around like 25 or 30%. So they've really increased that number significantly, but there's still 25% of patients that even going through all that genetic testing still don't get an answer. So that's the 25% we have to figure out. Yeah.
Do all babies born in this community
So to your first question, do the babies in the community participate in newborn screening? Yes, the majority of them do. And that's because there's been a lot of outreach to the midwives about the importance of newborn screening and offering it. So most of them will get the first screen done. In Delaware, we do a 2-tiered screening. A lot of them will not get the second screen, but they'll get the first screen done. Do they see a pediatrician? The better majority do not. Most of them do not seek primary care. The reason being is that they just don't feel like it's necessary. So a lot of their family members never sought primary care, and so it's just the idea that they don't need it. Now, if their child was sick, they would absolutely seek care quickly, but it's not something that they see as necessary based upon history tells them that most people, when you're well, you don't need to go to the doctor. Yeah. A lot of them do not. Yeah, immunizations in isolated communities such as the Anabaptist is a hot button topic, and it's something that we've talked to the state public health department a lot about because there's been outbreaks of like whooping cough within the Dover Amish, and so it's important to us to try to vaccinate as best as we can. When we've met with the bishops to talk about our work and talk about this research, we brought up the topic of immunizations and whether or not their communities, their church districts would be interested in that. And unfortunately, it's met with a lot of resistance. A lot of them are not truly in favor of it. Again, partially because of the reason that they said, well, we never had vaccines and we did okay. And because they just also feel like it's just something that's not necessary. So that's been something that we do wish that we could make an improvement on a little bit, but it's been difficult. Cool. Thank you guys very much. Appreciate it.